loss of the beta cells. So our company, ViaCyte, is really focused on how can we really cure this disease, or at least transform the way this disease is managed. And I'm going to tell you more about that tonight. But just a little bit of history. So for all the people in this room that have type 1 diabetes, and I know there's a few of you, I'm sure you're aware that if you had been born 100 years ago, you'd be dead. Okay? I mean, 100 years ago, type 1 diabetes was a fatal disease. And that was before insulin was discovered by Banting and Bess up in Toronto. They received the Nobel Prize in 1923 for discovering insulin and its effect on blood glucose levels. So that really transformed diabetes from a fatal disease into a chronic disease. Okay? And now we kind of fast forward 100 years, okay, since that discovery, a little less than 100 years, but about 100 years since that discovery of insulin. And the reality is, we're still treating disease exactly the same way we did 100 years ago. Okay, we're measuring blood glucose and we're administering insulin. Okay, and it keeps people alive, but it is not ideal by any stretch of the imagination. Part of it is the whole quality of life issue, and I think we've heard people asking about that, and pumps and such certainly have impacts on the quality of life. But the other thing is there's been studies like the ViaCyte study in the UK and such that have looked at very careful control of blood glucose with very diligent insulin monitoring, or glucose monitoring and insulin administration. And what it's shown is even when the most diligent people in controlling their blood glucose levels still lose over a decade of life. And I hate to say that to those of you who have type 1 diabetes, but that's the average. Okay, and sorry about that. So, and that's because of long-term complications associated with disease. And we don't know exactly why we have all those long-term complications, but there's different theories, like for instance, when insulin's produced normally in the body, there's also another protein that's produced called C-peptide, which we thought was a throwaway molecule that was just part of the production of insulin. Now there's work that shows that C-peptide might have impacts on microvascular disease. So if you don't have the C-peptide being produced, you can have complications with that. And there's a lot of things like that. Metabolism is a very, very complex path, okay? And so there's a lot that goes into that. So there's still a tremendous amount of long-term complications associated with diabetes, no matter how good we are at controlling blood glucose. So we haven't really changed it in 100 years in terms of how it's done, of what we do, but certainly I think the advances that were talked about tonight by Dexcom and Tandem, which by the way we use their equipment in our clinical trials, is fantastic. It is really transformed the way patients with type 1 diabetes are controlling it, and I think there's more transformations on the way. However, I think everybody would agree if we could just essentially cure it or do away with having to worry about measuring blood glucose and administering insulin, that would be the ideal. And that's what we're trying to do. And it's a big job, okay? And it's a tough job. And we've been working as ViaCyte, as a company here in town that's been around for well over a decade working on this problem. In fact, if you trace it back, it goes back about 2 decades where a number of companies came together. And so we are really trying to find a way to do it. Well, and the way we're doing it is cell replacement therapy using stem cells. And if you think about it, this is the ideal disease for cell replacement therapy because the problem is the loss of the beta cell. So if we can replace that beta cell, then we can correct the disease potentially. So there's actually some very good evidence that this is an effective approach, and that's what's called under a protocol called the Edmonton Protocol that was also developed up in Canada. I don't know why Canada has all these things going on with diabetes, but they've done well with it. But the Edmonton Protocol is using cadaver islets. So basically we take pancreases from cadavers, from patients who've died. We harvest those pancreases. It takes about 2 to 3 pancreases to isolate enough islets to treat a single patient. But it's a very effective treatment. This is used in very high-risk patient population, often what's called hypoglycemic unaware. You were asking about hypoglycemia. Hypoglycemia is one of the real problems with type 1 diabetes. Type 1 diabetes leads to elevated blood glucose, but when you give insulin and try to control it, you often get lower glucose. If you've ever been around a type 1, they'll tell you, oh, I feel kind of off, I'm feeling a bit funky, and they'll check their blood sugar levels, and sure enough, they're getting low on blood sugar. They'll go take some candy bar, drink some orange juice or something, bring it back up. Hypoglycemic unaware patients don't get those physiologic signals. By the time they realize that they're hypoglycemic, they're already at a point where they need somebody else to help them correct it. So if they're by themselves, they're in really big trouble. Okay, so for these patients, they are candidates for this islet cell transplant. There's been over, I think it's up to about 1,200 patients that have been treated with cadaver islets today, and it's very effective. It is a cure. Okay, these patients will go now with go as long as 5 years or longer now with completely insulin independent, with A1Cs in the 6% range. So all of you with type 1 diabetes know what I mean by that. Okay, so under very good control. So it shows that if you could replace that beta cell, you could potentially cure the disease. The problem with the Edmonton protocol is twofold. One, there's a lack of pancreases out there. There's actually waiting lists. A lot of people get kidney transplants, also get pancreas transplants. There's a waiting list for those organs. So that's one of the problems. There's a lack of material in order to treat the patients. The second issue is it requires, like other organ transplants, kidney transplants or heart transplants, you need continuous immune suppression for the life of the patient. That's gotten a lot better, okay, and the side effects from that immune suppression are not what it used to be. It's better. For the high-risk patients, that's a great trade-off. They're more than happy to take the trade-off of immune suppression for the risk of the type 1 diabetes that they have. But for a typical type 1 diabetic, that's not necessarily a great trade-off. So those are the things that limit the use of the cadaver islets. So our company has been focusing on how can we adjust that. And there's two technologies that we've developed, what we call platform technologies. One is using stem cells. How many here know what a stem cell is? Okay, probably most everybody. So what's unique about a stem cell, particularly the type of stem cell we use, which we use an embryonic stem cell, is it's what's called a pluripotent cell. It has the potential to become any other cell in the body. Okay? What's also unique about a stem cell is they are immortal. Okay? So we can take a single stem cell line and we can produce from that all the cells we need to treat every patient with type 1 diabetes in the world. Okay? From a single embryo that was used to produce an embryonic stem cell line. So we start with an embryonic stem cell. We bank those. We have working or master cell banks of these in multiple locations, by the way, so that if something happened to one of them, we're not out of luck. And then we take from that and we build working cell banks, and then we use that in the manufacturing process to go from that embryonic stem cell up to what we call a pancreatic progenitor cell. So this is a cell that has now been differentiated, so it's gone through down a path that it would normally happen in a developing organ, like a developing pancreas. And it's at a point where it's destined to go on to become an islet. Okay? Beta cells, alpha cells, delta cells, the full gamut of what you see in an endocrine pancreas. So that gives us an unlimited source of cells. Okay, so that addresses one issue that there is with the cadaver islets. And we actually have two products we're developing. One product we call PEC Direct. That actually is using those cells directly, similar to what's done under the Edmonton protocol, although we're doing it in a device under the skin. And that still requires immune suppression, but it gives us an unlimited supply of cells. So this is being developed for the high-risk patient population. But then we have a much more challenging but really exciting product that we're developing, which is called we're now calling PEC NCAP. We used to call it BCO1, but we just recently changed the name to PEC NCAP. And what PEC NCAP is the cells, but the second platform we have is a device, okay, that protects those cells against the immune system. And these guys had great technology toys that they could show and pass around and everything. So I don't have that. I have a pizza box and I have a flask. Okay, full of hot water. But I often describe PEC Direct, and I'm gonna do something. We're gonna pretend this flask is our body, okay? This is the blood or whatever you want. These are immune cells, okay? So these are cells that would attack our cells that we're putting in and destroy them, okay, if we were to implant these pancreatic progenitor cells that we've developed. So we need a device to protect those. And this is the actual device, and I almost would like to pass this around, but I've passed it around before and it didn't come back, and this is proprietary. So until I get it back, nobody gets to leave the room. But I'm going to pass this around. This is the device that we actually use. It's a very thin, very flexible device. It has what's called a semi-permeable membrane, which means that it has a membrane that allows the free flow of nutrients and oxygen. Proteins like insulin and such can go across it, but what can't get across the membrane are cells. And so I'm going to drop this in. So this is my mock-up of that device, because when I was thinking about what I could use as a visual in this, I often describe this device as a tea bag. So what you can't see is inside there's a tea bag. So hopefully that's going to brew some tea for us. But like a tea bag, let me go ahead and pass this device around. So, like a tea bag, which we got here, the cells are in that semi-permeable membrane, okay, in that device, and all the insulin and things can flow through that. But like tea, the tea stays in the bag, and that's what we need to have happen with our product. And more importantly, all these immune cells, the T cells and B cells that are part of the adaptive immune system that would destroy these cells, and the reason it would destroy these cells is these are human cells that we're putting in. The pancreatic progenitor cells that come from a embryonic stem cell line, a human embryonic stem cell line, but they're not your stem cells. They're not the patient's stem cells, okay? And therefore, your body would react to try to reject those cells if we put them in unprotected. And so this gives us the potential to have a product that we can implant into a patient, and it will control the disease, not be rejected, give us an unlimited supply of the cells, and be protected against the immune system. So I don't know, is this, my technology may not be working so good, but the tea bag is supposedly going to be putting out the insulin that is protected. But anyhow, so we've been developing this for quite some time, both these products. We are actually in the clinical trials right now with the PEC NCAP, the one that is really the kind of the knock it over the fence product. We're getting ready to start clinical trials on the PEC Direct, which is for the high-risk patient population. Currently, the trials we're running are at the early stage. This is unlike a typical drug where you would put that drug into a patient and you would get some result out. First, we've got to get these cells to survive the transplantation. If you think about it, when we have these cells in the manufacturing, before we implant them, they're living in an incubator at a nice warm temperature. We're bubbling oxygen through them. They've got lots of nutrients and all. They're having a great life. Okay, they're like you guys having a beer right now. They're kicked back. Okay, we then grab them, throw them into a device, and we throw them into World War III. Okay, we've made a surgical incision. We've made a pocket under the skin, and we've implanted these cells into that pocket. The body is reacting violently to those cells. It's hypoxic. There's a lack of oxygen because there's no vascular network on them when we first put them in. So we need to make sure these cells can survive that initial implant. And so the first part of this trial is all about that. We're giving subtherapeutic doses, and we're actually, this may be too small even to see, but this is the same, essentially the same as what I'm passing around. but it's a really small device that contains the cells, and we call this a sentinel. And the patients are real heroes right now. The patients are participating in this trial because they're getting subtherapeutic doses of the cells, so they're not going to get a therapeutic benefit from our treatment. But they're getting 6 of these put in different locations and then pulled out at different times. So they might come back 4 weeks later, we might pull 1 or 2 of them out. And then we look at these histologically, and we look to see, are the cells surviving? Are they vascularizing? Are they becoming beta cells and other cells? Are they producing insulin? And so this is how we can initially determine what are the conditions we need to have to make sure that this is a successful product. And it's a long process because it takes 4 to 8 to 12 weeks before we can pull these out and see what's going on. And then we make some changes based on what we learned, and then 8 to 12 weeks later we find out if that did anything. So we've done over 12 patients now, and we are learning a tremendous amount. I would say, we can't talk in detail about it, but what I would say is we're making good progress. It's a learning curve for sure, and it's not only giving us information for this product, but also giving us information that will help us with the Pet Direct product, the one for the high-risk patient population. So we're tremendously excited. The team at ViaCyte is absolutely dedicated to finding a transformative therapy, what we call a functional cure for type 1 diabetes. We also believe that this will be an effective treatment for insulin-requiring type 2 diabetics, which represents about 30% of the type 2 diabetic population and growing. So we believe it will be a therapy for that. In that case, though, that won't be a cure. It's almost unfortunate that type 2 diabetes and type 1 diabetes have the same name. They're really very different diseases. Okay? What causes type 1 is autoimmune disease. What causes type 2 is a whole raft of things, including lifestyle and various other things. So they're very different diseases, but they both impact glucose. They both are regulated by insulin, but in very different fashions. So we do think this will have applications for the type 2, but not as a cure, but as an adjunct therapy for those patients. So we're continuing with this clinical trial. Once we have optimized and we're convinced that the engraftment is occurring the way we need it to, then we would move on to a second group of patients where we would put in a therapeutic dose of the cells, and then we'd look for efficacy. And efficacy is pretty straightforward. We enroll patients that have no ability to produce insulin on their own, that are called C-peptide negative. As I mentioned before, C-peptide is a breakdown molecule when insulin is produced. And so we can measure and look to see if C-peptide is produced after we treat them, and if it is, it's doing what we expect it to do. So that's, in a nutshell, what we're doing at ViaCyte. The company is about 60 people, about 65 people now, and we're based here in San Diego, and we're going to continue working on this until we find that cure.